everybody, I have Dr. Kevin Bateman, Vice President of Merck, on the line. He is not only a colleague, but a friend of mine, and so honored to have him on the show. And today we're going to be talking about how to empower patients with the virtual clinic with Dr. Kevin Bateman. And we're going to talk about other things too, about his history and our history and lots of other things. But again, it always has that theme of never be outworked. So welcome to the Secrets of Success podcast with Dr. Shane Needham. That's me, where our theme is never be outworked. And Kevin just absolutely is an example of that never be outworked theme. We'll hear about his life and his history. And obviously you don't get into a position like Kevin's been in without having a mindset of never being outworked. Lots of late nights, lots of flights across the world, lots of 3 hours worth of sleep and getting up and giving a presentation and then getting on an airplane and leaving again. I've heard those stories. I've seen him in action doing those stories. Again, Shane Needham here with the Secrets of Success podcast. Theme is never be outworked. Follow me on Facebook. Follow me on Instagram. Follow me on LinkedIn. I am so excited. And I really have to thank all my listeners and followers. In the last a little over a month, I have had over 60,000 views on some of my social media. And that is incredible. Incredible. Go check out my website at drsneedham.com. Go check out my YouTube channel. I have all my previously recorded podcasts on there. This is the 15th podcast. I'm super excited to have Kevin as a guest. Check out my previously recorded podcast on Spotify, on iTunes, on SoundCloud. We are taking callers today. You'll see that banner that's streaming. So when it's appropriate, we'll take a caller. I might just automatically put you on a mute, but you'll be on hold and we'll get to you when we can and when it's appropriate. That number is 877-995-2111. And I've had the last couple of weeks some awesome scientists on the show. We've talked about mass spectrometry. We've talked about chromatography. And it really is so exciting for me to finally bring social media and a voice of scientists to the global community of a lot of people that aren't scientists. And what I've been finding is because somebody like Kevin or myself, I guess we're doctors or we're PhDs, whatever it is, a lot of people feel that we're strange. Now that's probably true. We are strange. I will agree with that, but we're not untouchable. And it doesn't mean that we can't communicate with the normal communities and try to use jargon that will get the correct message out to them, including the science. But what's important is making sure they have the right information and not misinformation. We really realized this during the pandemic, that we've seen so much information come out. And is it true? Is it false? And you don't really know. And science is the one that will figure out if there's truth or not. And what's true now may not be true a year from now, because we'll show more science. And so I'm so excited to get that voice out to our communities and be part of that social media. The future, I don't believe, is getting scientific information out there in white papers or conferences for the general public. It's going to be formats and venues like this. And so, Kevin, I'm so excited to have you on the show. Thank you for being a guest. I've always loved your presentations. I've always enjoyed your presentations, your science, your papers, our interactions at conferences and such. And so thank you very much for being on the show, and tell us your story. So tell us your story. We want to hear about you, and go back as far as you think you need to give us a good history of how you became a vice president, why that might have started 30 years ago, why you got a PhD, why you like science, all those types of things. Tell us about that. Start. You're on.
Shane, no, it's a pleasure to be here. Thank you very much for the opportunity to come on and talk to you. It's like you and I having a conversation, like we're at a meeting somewhere.
Absolutely. It's great.
How long do we have? Like 5 or 6 hours to go through my whole...
Hey, if that's what you want to take, we can take 5 or 6 hours. If you want to start like, I was born in 19...
My father met my wife, my mother. Right? Yeah.
Yes.
Yeah. No, so I'll go. So I'll start. I'm Canadian, right?
I'm sorry. Actually, that's great.
I'll start with that. Living in the U.S. now. I live just outside of Philly. Been down here almost 10 years now.
Wow.
Yeah. But so back to Canada. I'm an East Coast boy from Nova Scotia, Halifax, Nova Scotia.
Can you, especially for people out west, and a lot of people are out west watching, they don't even know where that's at, Kevin. I know because I have friends from Canada in that area.
You hit Maine, and then you like turn right and keep going, and go, and then then you get to Nova Scotia. So it's
And isn't that near Prince Edward Island as well?
Island, yeah, it's another province just on the north side of Nova Scotia, and New Brunswick and Newfoundland. That sort of makes up the Atlantic provinces. Right?
So Newfoundland, I remember listening to some of my Canadian friends, and I learned all about Newfoundland. And you call them Newfies, right?
Yeah, that's a good derogatory term for our.
And it's like, this is what somebody said. I don't know if it's true or not still to this day, but when you go to Newfoundland, you like step back in time by like 200 years. Is that still true?
No, it's a pretty modern place. Although if you want to go see some of the original Viking settlements in North America, they're there in Newfoundland, right? So
that's cool. I did not know that. That's really cool. Okay, maybe as a Canadian, you know this history too. Why do they have a time zone that's a half an hour different?
Because it's Newfoundland. But it is pretty far out. Like
it is.
It's pretty far out there, right? Like it's there's only a couple ways to get there. There's a ferry that goes from Nova Scotia to Newfoundland. It's a 24-hour ferry, right? It's not like a small ferry ride, right?
Is that right? Oh my goodness. But you can drive there if you drive up through Canada, can't you?
No. Newfoundland, Labrador, Newfoundland and Labrador are, so Labrador is on the north side of the province of Quebec. Newfoundland, the island, is out in the Atlantic Ocean, and there's no way to get there unless you take the ferry or you fly.
I did not know that. Oh my goodness. Okay. That explains a lot more. It's very isolated.
Yeah. And it's a big island, right?
Wow, yeah, definitely. That's why I thought it was just kind of mainland, because it was so big. So, okay. So you're from Nova Scotia, from Halifax.
So my dad was Royal Canadian Mounted Police, a Mountie, you know, the red surge jacket. So as a kid, we moved growing up quite a bit, right? That was part of being a Mountie. So we moved around, which is, I think, something that's kind of formative for you as a kid, right? You learn to adapt, right? You go and have to experience new things, make new friends, entertain yourself because you don't have friends in a new place. So I think moving was a formative experience for me growing up, and then I lived in different parts of Canada too. So I lived in Ottawa, which is the capital of Canada, then moved back to Nova Scotia, and then went to university. I don't know, and I think like most kids, you're not really sure, like an 18-year-old, what do you want to do with your life? Some people are very focused. I started in undergrad in sciences, did a couple of years, still not really sure what I wanted to do. And then I had an opportunity, and I moved to England for a couple of years, so about a year and a half.
Really? Oh, wow. Okay.
My parents got moved there, so I took some time off, and that was, like, again, a learning experience. You're not in school, but you're still learning, right? School, there's lots of learning to be had outside of school. So I wasn't a good student when I was my first go around at university. So I think I had to mature. I grew up working in England for a year, realizing a bit more about life, and also realizing I didn't want to have a routine office job, because that's what I was doing for a year. It's like, oh man, there's got to be more to life than working in an office for 8 hours a day. And so when I went back to university, I treated university like a job, right? It's like, because university, it's very different than high school, right? It's like, you're not spoon-fed. The profs are like, okay, you're here, your job is to learn, here's the content, go figure it out, right? And so some kids can adapt to that and others can't. And so I treated it when I went back like a job, 9 to 5. If I only had classes 2 hours a day, I'd still work the rest of the day on other things, right? And so once you have that mindset, that attitude, like put the hours in, right? The secrets of work, do the work. then I became an A student, right? And still didn't know. I focused. My major was chemistry. Why chemistry? I don't know. I liked it. It was not as hard as physics and more. What's the right word? More, I guess, more solid, more than biology. Biology was kind of nebulous. Biology was always like the study of exceptions.
Yeah.
It was much like math, and chemistry sort of bridged between those two. So I finished my undergrad, and I also did an education degree because I thought I wanted to become a teacher. Right?
Wow, interesting.
I was going to become a teacher because basically because I didn't know what I wanted to do. So I did a teaching degree at the same time as I was finishing my undergrad chemistry degree. I did all the student teaching. I did some teaching of like middle school, junior high in Canada, like grades 7, 8. So
very interesting.
A tough crowd, right?
Definitely.
And then I did a stint in a high school teaching some honors chemistry. That was good, much more focused students. And then, but I thought that if I was going to become a teacher, I should know more about the science. I wanted to go do like a master's degree and learn more about research. And then there was a program at a university in Canada that did a co-op master's. So you do part of it was classes and research, and the other part was going to work in industry. And so I said, oh, that'd be good. I'll have that good experience to become a teacher. So that I went and I did my first semester. I took classes, and then that was September to January. And then in January, for the next eight months, I worked at a national research lab in Canada, so a government-funded research lab in Canada, and that's where I first came across mass spectrometry. Right?
Now, was that working with Bob? I'm forgetting.
Yeah.
Who was it, Bob?
Boyd.
Yeah, exactly. Yeah, okay.
And Steve Pleasance and.
Wow. So was Bob Boyd? Is that who you did your graduate work with as well?
No, because he had left. He was at the University of Guelph in Ontario, where I started my master's, but he had left and joined the NRC lab in Halifax, and he had actually set up the co-op program. So it was good.
That's awesome. Okay.
So I started doing research, and I was like, wow, why would I teach if I can do this research? And so
absolutely.
I finished my 8-month work term at the lab, the National Research Council lab, and I had to go back to Waterloo University, where I did my master's, and finished my master's in a year, and then went back to Halifax to this National Research Council lab. Because mass spectrometry, the technique that's where I first got introduced to it. I worked on, it was a technical SciEx API 3, but it was serial number 3 out of SciEx.
Well, Kevin, I'll tell you the story. So you might know this story. That's pretty cool, actually. So whose lab was that? Bob Boyd's lab? Yeah,
that was the Halifax lab, because they collaborated with SciEx. So serial number 1 was at SciEx. Serial number 2, I think, was at Iser, and serial number 3 was.
Now, here's the actual story. So I worked on serial number 2 at Pfizer. Right?
Yeah.
Here's the actual story. This is what Tom Covey was saying. Is that was actually serial number 1. They didn't have enough money to make serial number 2. They just labeled it number 2. So once Pfizer bought it, I think back then it was a lot. They're more than they cost now, actually. But they bought that one, and then they actually made serial number 2. Now that's the story. Is it true? I don't know, but that's awesome, you guys. I didn't know that. I wouldn't have predicted that's where the third one had went. Actually, I would have predicted it probably went into pharma or something like that. So that's really cool. Those were great instruments, by the way. They were amazing.
So it was falling out that changed the course of what I wanted to do. Right? I didn't know. I was my supervisor for my master's was an elemental analysis guy. So I was doing inductive through coupled plasma stuff. But then when I started doing this electrospray mass spec, I'm like, this is going to be big, right? and so that was like 91. That's
wow. Wow. That's awesome. That is so cool. And so what university was that you were doing your PhD there?
University in Halifax is the name. Yeah. So, right? And so that's kind of funny too. And so I worked on my PhD, and it was interesting because, those systems, you had to recycle them every night. So they, 10-hour recycle and 48-hour on the weekend. And getting time on it was precious. So when you got time on it, you basically worked 24 hours,
absolutely,
as many hours as you could, and loaded up as many samples, because you wouldn't get on it again for another month.
Right. Yeah. The instruments back then, you only had, let's say, a 12-hour window to work on them. Yeah. And then you had to, let's just say for the non-scientist, put them at rest for 10 hours. Otherwise, it wouldn't work well. And so 10 or 12 hours. And so you had these 12-hour cycles. So you would have to get on them and get things done.
Tired, right? You got to give us some rest. Right?
Yeah, it was crucial, I think, to understanding how to be very efficient and productive. And so now, did you say you were at University of Waterloo?
Yes. Yeah. That was
I probably can't pronounce the gentleman's name correctly, but he's the solid phase micro extraction guy, Janice. Yeah. Did you work with him at all?
No, I worked this other guy, Vasily Karanasius. He's still there. They're both still there.
Wow. Yeah. Interesting. What a small world. And that's what I love to hear about the stories. And then we're talking about science, but is just what I've realized over the course of my career is the science has been so cool. We've watched it all develop. You've watched it develop. You've been a big part of it, developing it. We'll talk about that in a minute, but just meeting the people and hearing their stories. And what I like about your story, Kevin, is it's very similar to a lot of other people's stories. And I was kind of the same way, as far as didn't want to study biology. So it's like back then it was naming plants and animals, and there's all these exceptions, but I really loved chemistry. But it sounds like you were first generation college educated. Is that true?
Yeah. My dad never finished high school. He went back and got like an equivalency certificate later, but he was adamant that all his kids would go to university. So I'm one of four, like an older brother, an older sister, and a younger sister. So
that's awesome. Yeah. What a great story. I'm the same way. First generation college educated. My parents were very blue collar, and they worked really hard to make sure we could have the best life that we could. But we were certainly lower middle class, probably.
Yeah. You don't know if you're poor or rich or whatever, right?
No.
We had summer vacations. We'd go camping, right? That was
that's what we would do. It's funny. I look back and I think I never thought we were poor, because we always ate. We always had, it wasn't extravagant food. And when I say never, I mean,
creative cook, and she could turn hamburger and macaroni into a special meal, right?
Exactly. And we would, never go out to dinner. I mean, you wouldn't go to Pizza Hut or McDonald's. It was like, you just didn't do that. And so what a great story. So you finished your PhD, and what was your dissertation, or basically your PhD research? What was that based on? Mass spectrometry.
Mass spectrometry, coupling, capillary zone electrophoresis. So, a very specialized separation technique.
Oh, I did not know. So you worked a lot with capillary electrophoresis.
Yeah.
Oh, wow. Interesting. Okay.
And glycoproteins and glycopeptides. So proteins that have sugars attached to them. So
that's really cool. Well, we might talk a little bit later about capillary electrophoresis. I'm still fascinated by it. I still think it has a niche, but it has lots of issues.
Yeah.
I think there's a lot of cool things you can do with it. So maybe we'll get to that later. I didn't know that was your PhD research and your dissertation. So what I like about CE, again, we won't get into it too much, is I think it's the future, because you can miniaturize it much more so than a liquid like high performance of chromatography. Anyway, so those are some details. So that's awesome. So you graduate, you get your PhD. Yeah. What's next? You're ready for your...
I had a postdoc. I started, because I didn't know, okay, I get a PhD. Now what do? Oh, I get a postdoc. And so I had a postdoc all lined up to go to Dick Smith's lab out in Pacific Northwest National Lab.
That's about 2 hours away from me right now, actually.
Yeah, but I never made it there, right? And so what happened was a guy that did his PhD in the same lab as me had gone to work at Merck in Montreal, Merck Frost, as a postdoc, industrial postdoc. And him and his wife decided that they didn't want to live in Montreal, just, for personal reasons. They were from Ireland originally. So his wife was in vet school, and, she would have had a hard time working in Quebec as an Irish person not speaking French. So they wanted to go to an English speaking province. So he said, "Hey, Kevin, you should come work here, or you should, it's a great place to work." So, and it's funny, like, finish your PhD, you're hot, well, I won't say the word, but yeah, hot poop, right? You're hot stuff, right? And and I had a postdoc lined up, so I went and interviewed at Merck Frost with the attitude, "Hey, if you like me, great. If you don't, so what? I have a postdoc." And so I interviewed, and it was a good interview, really good people, like that mixture of science and biology and analytical research, and so they offered me the job. And so I went to Bob Boyd, so he's a Scottish guy, mentor, and I said, "Bob, what do you think I should do? I have this postdoc with Dick Smith. I have this job offer from Merck Frost. What do you think?" And he looks at me and says, "Tell me, lad, how old are you?" Right? And I go, "Oh, well, I had just turned 32, right? So I was old because I skipped some school, and he looks at me, he goes, "Take the job, lad." Yeah, that's good advice. I will do that, right?
I think that probably was good advice. Yeah. Well,
yeah.
That
was 97. Yeah, so now you can do the math and figure out how old I am. But so 97, September 97, went to Montreal and started working at Merck Frost. And it was a great environment, great place. One of the most successful drug discovery labs in the world ever, right? I learned a lot about drug discovery and process, and it was very, like, coming out of grad school, you're working crazy hours all the time, right? So what I didn't tell you is that I got married just before I started my PhD. My first daughter was born during my PhD. I used to walk to school carrying my daughter in like a child carrier backpack and drop her off at daycare. I could see the daycare from my lab window, and then go home at night with her, have dinner, bathe her, put her to bed, and then go back to the lab to work. Yeah, and so moved to Montreal, and you go to the lab, and there's people there all hours, weekends, and like, oh, well, that's what I was doing in grad school, so that's what it's like working. So that was just the work ethic. You just did the job. It was, and so many smart people, so driven and focused on, we must find the best possible molecule to put into clinical development. And that was the singular focus. They didn't care about arbitrary timelines. Like, if this is not going to be a drug, we're not going to put it forward, right? Yeah. So it was a great environment.
Was there, just curious, was there high throughput screening in that?
We had a screening capability, not as big as the one in the US, but a lot of, smaller, like, 50,000 compound type screening, not millions of compound screening. So we had all that capability. It was discovery from target ID all the way through to candidate selection. So it was.
That is so cool. So, man, I just love your story, and I've never heard it to this detail. I really appreciate you sharing it, and I'm sure our listeners do too. And so it kind of makes me wonder, and just ask you the question, so obviously you knew how to work hard, whether it be taking, your daughter to daycare, dropping her off and going to work in a lab and working long nights and all that. Where do you think you learned your work ethic?
Yeah, I know. That's a good question. And I don't think I see it as work, right? I think it's, if you're really passionate about what you're doing, you don't see it as work. It wasn't like, oh man, I have to go do this. It's like, I want to go do it, right? And that's, what I've told my kids, and my younger daughter going, like, she had to write a letter for grad school and about why she wanted to go to grad school. And she wrote like, oh, my dad told me if you find something you really love doing, you never have to work a day in your life, right? And that's sort of the attitude. It's like, okay, hey, I want to go there because I want, like, this, the science it's fun. It's like solving problems that no one solved before or seeing things that no one has seen before. So how does that work?
It is fun. So what a great mindset. So you don't consider it work. I feel the same exact way. And I think that, there's a really good saying that some people say, follow your passion. And there's some truth in that. I like to use the term, be passionate about what you do.
Yeah.
And I think there's a difference there, because if we really followed our passions, we might be playing guitar on the street, but we're not going to be able to really make a huge difference necessarily. And I don't want to take away anybody's dreams. Dream big, but whatever you do, I've had some crazy jobs, like you have. I've worked in factories. I've been a janitor. Worked on farms. I treated every one of those things like I loved them.
Yeah.
I think that's a big difference. And I can see that in you. If you guys ever get a chance to, I think, Kevin, didn't you do a TEDx talk?
Yes. Well, for Merck in Merck one. Yeah. Went through all the training. It was pretty cool.
So Google him. He is a great, Google him for a TEDx talk.
Inside of Merck, right?
Oh, is that right? It wasn't extra. They didn't publish it outside? Okay. Well, anyway, Google Kevin anyway. You might see some YouTube videos or something.
There's a CSA video.
Oh, is that right?
Yeah.
Okay. So he's great. You can tell he loves what he does. Look at him, just how relaxed he is and how much he loves it. Always positive, always passionate about what he does. So thanks, Kevin. That's easy. So the way to never be outworked is to not treat it as work. And that's how you do it. So really, really good job. So we were talking offline the last couple of days over the weekend about how we met. And I think I remember now. He reminded me. So I like to kind of connect everybody. So how do you remember how we first met, Kevin? When was that?
I remember a meeting in Baltimore, right? At a bar, like, because we go to a lot of meetings and you meet a lot of people, right? And I think your memory of me might have been like at CPSA, what you said before, but that, yeah, for me, it was a more intimate, meeting. Yeah. So we went out for bar and had like beers and chicken wings or like that. It was just a small group of people, right? And we hadn't really met or spoke a lot, but I remember like, oh, who's that big muscle guy?
Who's that big muscle guy? Oh, well, thanks. Okay, big muscle guy. Well, that sounds good. I do remember that time. And wasn't that about the time you were leaving Merck Frost?
It was, yeah, because Merck closed at the end of 2010, right?
Yeah. So that's what I remember. I remember the conversations like
in that transition period. Yeah.
Yeah. I mean, I'll be honest with you. Typical ethnocentric American kid from the USA is like, I was like, oh, is Merck Frost part of Merck? I didn't know.
Most people didn't know. Right?
No, a lot of people didn't. And there's still some European Mercs that are kind of related, but not really related. So it is kind of confusing. But I was like, I remember seeing all your names in the white papers and stuff you were doing. I'm like, oh, wow, that's Kevin Bateman. Pretty cool. And so anyway, and glad you came to the States and glad for all the research and all the impact that you've made in science. And so we've heard about how we met. We heard about your science history. We've heard about all those great things. And then you go to Merck. You go to Merck where you're at now.
Yeah.
And talk a little bit about before we get into clinical trials and what those are and how we can talk about those. Talk about you came to Merck. You're still there. You're vice president now. What's your role now? What has it been, and what is your role now? Yeah,
so I'll start with the transition because that was important, right? So Merck Frost shut down, whatever the reasons is, we don't need to get into. And when I was there, I was focused mainly in discovery. And so now I moved to the US. I could have stayed in Canada. I had an opportunity, but I decided, and it goes back, one of the reasons was I wanted my kids to understand and experience moving, right? Wow. And so part of the motivation was, and we moved them mid-year, right, at Christmas. We took them to school, moved them to the States, put them in a new school. And that was, traumatic for them, but man, they're so much stronger for it now. They've learned, that moving and meeting new people is not, it's part of who they are now.
Yeah.
But when I came to Merck in the States, I started working in the regulated space. So, we have discovery where we, understand the biology and try to find a molecule that we hope can become a drug. And then in the regulated space, we start doing clinical trials and testing them. And so I started working in that space and learning about regulated trials. But my job at Merck over the years, it's transitioned from working on very specific projects as an analyst and analyzing samples to being more involved in the technologies and approaches that we've used. And that was part of the transition as well. Just before Merck Frost shut down, I took on a more global role about the analytical technologies all across the labs. And so I got to go visit all the Merck labs around the world and interact with the people there. And then when I moved to the States, I continued that. My job and has been to push, push the labs to improve, to take in new capabilities, to take on new approaches. We become very, not complacent. We become, set in our ways. It's human nature. We do things a certain way. They're working, so why should we change, right? And for some things, there is absolutely no value in changing, right? So we don't change just for the sake of changing. You have to identify, why this might add value. And so that's sort of been my job. Where are we going to be, 5 years, 10 years down the road? What are the capabilities we should be putting in place? It's tough, right? People, some people love change. Some people don't want change at all, right? And then in the regulated space, that is even 10 times harder, because the regulated space, you have a lot of regulations, you have government oversight for a lot of the activities. There's guidances that you have to follow. You get inspected. Companies will go out of business if they're caught breaking the rules, right? And so compliance and patient safety, because that, at the end of the day, is the most important thing. We don't want to put patients at risk, so you have to be very compliant in what you do. So changing things, you have to really investigate carefully. Are you, what you're changing, how does it impact the quality of what you're doing? Yeah. So, and then I just took on a few different jobs, areas where I thought, hey, we need to grow here, like protein mass spectrometry. So I left a group that I've been running and said, okay, I'm going to go off and start a new group. And I built that up. Just the recognition for that over time. Merck has a research ladder and a management ladder, different ladders. So I'm on the research ladder, and it goes from R1 through R8. And so my position is R8, and it's associate vice president, not full vice president. It's associate vice president.
Well, congratulations.
It's the top of the research ladder at Merck, which is, I think it's pretty cool to make it there. The first analytical chemist to make it to the top of the research ladder.
Wow.
It's interacting with colleagues at other companies it's been good. They're so happy for me to have reached that, because then they see it as an opening for them as well, right? Someone has
to. Yeah, wow, Kevin, that is a great story. And he has a dream job in the industry. He really does. I mean, I've watched what he does, and he gets to work with cool tools and design things. Now, here's what I can tell you. I don't know about the internal discussions at Merck, but here's what I can tell you what Kevin's not afraid of. And I think this is important, is there's a lot of people in science and even in the world that they don't say things because they're afraid. They're afraid of maybe what somebody else will think of them, or what will think of their science, or they might think they're stupid, or it's a dumb idea. Kevin is not afraid to say, this is how it should work, and here's why, here's the data. And that's not always the safest position. A lot of times it's you could just follow everybody else and say, hey, I'm just going to do my job. But look at the position he's in now. And he gets to live his dream because he was willing to say, to probably take a risk and sometimes say some things that are going to change up things a little bit, and that a lot of people are afraid of. but it's better for the organization. It's turned out really well for him. So congratulations, Kevin. It's great to hear. It's great to hear the position you're in, and I think that's awesome. And you're only going to continue to grow. So congratulations. Okay, so let's talk about clinical trials. At a high level, people might know what they are, but the general public, they probably don't. So explain what clinical trials are for the non-scientists and why are they important?
Yeah, so at a very high level, clinical trial is where we first test a new potential drug in humans, right? And so that's really what clinical trials are. But there's several stages to that. It's a very long process. There's a lot of data that goes in behind the decision that enables you to take a new potential therapy into humans, right? And by new, means new. Like this molecule, whether it's a small molecule or a big molecule, like a protein or a vaccine, never existed in the world before until the company made it, right? So what happens when you put this in a human? Patient safety is, I work for a pharmaceutical company. Our goal is to help people. Our goal is to provide medicines that, treat unmet medical needs. So it's like doctors, do no harm, right? And so there's a lot of years of work that goes in the discovery environment and then in the preclinical space to test safety. All of that data has to be understood and provided to regulators, and so they make the call on, yes, we agree that you can proceed into human testing. And then, so phase 1 human testing is very small, healthy males, and you're putting in at really, really low doses, well below what might be therapeutically active, just to start getting any assessment of is there any safety issues, right? So phase 1, single dose, right? Low dose, and then you start doing dose, single ascending dose. So you start increasing the dose up to some point. You might have an idea of what side effect profile you might see. There's all sorts of different ones that get looked at. Maybe it causes heart rate to increase, and you start to, we start to see that, we need to back off the dose. Then you start going into multi-dose testing, because as you get more and more doses, does it accumulate and cause any issues? So very cautious, very early, looking at understanding safety. Nothing to do with whether it works or not for the most part, depending on the disease state. And once you get through that testing, you get sort of an idea of how much dose gives how much drug in a person. You go into phase 2. And phase 2A, early, but now you're talking patient population. So you start doing some early testing, and hopefully, depending on the disease state, you might be able to get a quick readout of whether it's working, right? If diabetes, and looking at just glucose and glucose tolerance tests and things like that, you can maybe get an assessment of what's going on. So then you have a small efficacy test, and then you expand. And if that looks good, you're trying to figure out what dose is going to work. And if you get through that, then you go to phase 3, and that's much bigger studies, depending again on the disease area. And you test in a bigger population. But bigger, it might be 5,000, patients, 30,000. And so it sounds like a lot, and it is a lot of people, and it's several years depending on what the indication is, but it's still not the real world. And so we can never know exactly what's going to happen until we get out into the population, right? And so that's why you see things like, we didn't know there's an adverse drug reaction, just because once it's out in millions of people, something that only happens in like 0.01% of people, you don't pick up in a phase 3 clinical study for the most part.
Right.
But they're very rare.
Yeah, that's the thing is there, all these therapeutics that you're going to be, ingesting or taking in whatever mode there you'll be for getting the dose, there are risks involved. There are definitely risks involved. I mean, there are, and part of a clinical trial, would you agree, Kevin, and all the data that goes with a submission to a government agency such as the FDA, it's all part of a risk benefit ratio. And does the benefit outweigh the risks? Is anything we do in life is going to have a risk.
Yeah, and so linked to the, like, for oncology, you have a much higher risk tolerance for giving something that might have a side effect profile, because guess what? If you don't treat them, they're going to die, right? Their cancer is going to progress. And so, and that's why we have a lot of, toxic chemotherapy agents that, people lose their hair because they're giving, but you're killing the cancer, right? And so we've seen a change in cancer therapy. But then if someone has high cholesterol, then the tolerance for a risk profile is low to none, right? There's good cholesterol medicines on the market that have been around for a long time. So trying to bring a new one on that space is just the bar is so high, right?
Right. Exactly. And so what are the, talk about what the current challenges are with clinics. I know this has been a space that you've been actively involved in and trying to empower the patients and trying to do what's better for the patients. And that's, whenever we do what's better for the patients, that's going to be better for all the communities. It's going to get more therapeutics or better treatments or ways to treat disease to more people. And that's what we want. And that's what our goals as scientists are in the healthcare industry. So what are some of the current challenges with clinics?
Yeah. Well, think about it. Enrolling in a clinical trial. Say, you're, out in rural America somewhere as a farmer, and you develop, cancer, and you want to enroll in a cancer clinical study. Where's the nearest clinical site? You're probably a couple hours drive away, right? And you want to enroll in a trial. You're gonna have to drive there 2 hours, check to see if you can enroll, and then maybe you have to go back like every week for some tests, right? 2 hours there, 2 hours back, half a day or more at the clinic. Do you have the time to give up that? And so we tend not to enroll people unless they're, desperate and or close to a clinical site, right? And so there's stats out there, you believe them or not, it's like what is it, like 30%, only about 30% of people in the US live within, 2 hours of a clinical site. But 90% of us live within, half an hour of a drugstore, like a pharmacy, right? And so there's opportunities to, and so that means you're enrolling based on the population that's around your clinical site, and so clinical sites, you may not get the diversity, whether it's in age or ethnicity or socioeconomic status, right? And because there's, enrolling in a clinical trial, you get reimbursed for costs, but if you have to take a bus, 2 hours to get to a clinical site, how likely are you, to enroll, right? And you can't give up the time from your job to go. So, being more patient-centric in clinical trials, I think, is an opportunity, and we're seeing it, especially in our current, pandemic, where traveling to a clinical site might be putting yourself at risk, right? Because you're being exposed out in the public. You're going to a health center where there might be people who are infected and being treated. So decentralizing and making clinical trials more amenable to remote type activities. Now, to be fair, there's some things you just have to go to a clinic to get done. If you need an MRI done, guess what? You have to go to where the MRI is, right? So if you need a tumor biopsy sample collected, then you have to go to where they can safely get a tumor biopsy. But there's a lot of other things, like it's an evolving space. We're calling them digital biomarkers, but just things like heart rate or motion, cough, right? Movement disorders, smartphone tap tests to look, and to measure cognition and things like that. All these things. Why do you have to go to a clinic to have a doctor do those types of assessments when they can be monitored all the time in real time? If you're walking around with a smartphone in your pocket, it can track your motion, right? Patches that go on people that monitor all sorts of physiological outcomes. So, yeah.
Well, it's great. You've talked about those challenges. I mean, obviously, as people watching and listening, you can tell that a clinical trial trying to get a therapeutic to the market, one of the actual, and it depends on what the indication is. That means what disease you're trying to treat. It can be very difficult to find people to even enroll in these studies. Okay. And so once you find how to enroll them, then you got to realize what's the cost. How efficient is it? How productive is it? How inefficient and unproductive is it getting people there? And that all increases the cost of these therapeutics. And I love telling a story. I was visiting a client. It was a smaller biotech firm, and they were sharing a facility in like a technology park, and they were sharing it with a hospital. And I remember sitting in the waiting room and watching this lady who wasn't elderly per se. I would say she was in her 50s or 60s. Okay. And she, had her head wrapped up. Probably she didn't have any hair. And she was getting out of a taxi cab and she was using a walker. And you could tell she didn't need to use a walker, but she was probably so weak because she was probably undergoing some kind of oncology treatment or cancer treatment. I don't know. And it's my understanding that she was coming to the clinic to get like a 72-hour blood draw to see what the levels of a therapeutic were in her blood. And that just seemed so inefficient, unproductive, and you know what? Uncomfortable for the patient. And so there has to be a better way with all the technologies we have. And so one of the things that Kevin has been working on is at-home sampling and virtual trials, and what does that space look like, and how important that has probably been during this global pandemic, because a lot of things were put on hold because people couldn't go to the hospital. So can you describe at-home sampling and virtual trials and what those are and how it helps the patient?
Yeah, so one area that I've actively been working in for a really long time, and it's kind of sad that it's been so long, but it is around blood collection in the clinic, and trying to make it more remote, so patient-centric. And a lot of it, there's different aspects of it. One is shrinking the volume, right? Normally when you go and get a blood draw, they're putting a needle in your arm and filling up a bunch of tubes with blood, labeling them and sending them off, right? But a lot of times we don't need that much blood to do the analysis we do for a clinical study, right? If we're measuring that 72-hour bleed for exposure, and by exposure, it's how much drug is there, that's a small amount. A few drops of blood could be used for doing that. And so that's what I've been working on. It's called microsampling, so really small samples. And it started with, for me, with animal studies, mice studies. Mice are really small. And can we just take a couple drops of blood from a mouse and use that? And so that started many years ago. But then taking that same concept to the clinic and making it convenient for patients to collect samples and small samples that we could use. And I first got exposed to at-home sampling even before I started working at Merck. When I was doing my PhD, when my first daughter was born, and I lived in Halifax. So a public health nurse came to our house and did a heel stick and collected dried blood samples from my daughter, because you do, screening of inborn errors of metabolism. They do that on all kids, everywhere, right? And so in Halifax, and when I was there at the time, they don't do it at the time of birth. They come up, a couple of weeks later to collect samples, right? And so I was like, wow. And so when I started at Merck, and we're doing like some mice studies with mice that were very expensive to buy because they were genetically modified. And it's like, wow, these are expensive. How can we cut down on how many mice we have to buy? And I thought about, hey, well, if we can take a drop of blood from my daughter and use that for measurement, can't we just take a drop of blood from a mouse and use that? And so, yeah. And then others, we published on that and presented on it. And others like Neil Spooner, Matt Barfield, and others at GSK. Chris Evans started like publishing on using it in clinical trials. And then Merck started pushing that. So I pushed it internally at Merck, and it's evolved. We had some hiccups along the way. We've learned a lot because there's so much upside to it. Imagine, right? That person you talked about didn't have to travel all the way to the clinic to get a blood draw when she's, not feeling too well, and we just sent the device home that they collected a sample and mailed it in. There's so much benefit to that.
It's so much benefit. And how does that relate? So at-home sampling, I absolutely will, and we'll get into my own personal story as well, because I think it's important, because sometimes people try to relate this to clinical trials and, oh, that doesn't apply to me, but I'll let you know how it does apply to you in just a minute. And Kevin kind of alluded to that with his own daughter.
Yeah.
But how are home sampling and virtual trials, how are those connected? Or can you describe those?
Okay. Well, I'm not sure what you mean by virtual trials, but
I want to know what you mean. Now, it doesn't matter what I mean. I want to know what you mean by virtual trials.
So I would say hybrid trials, not like virtual. I don't think it'll ever, we're not going to stop visits to the clinic, right? You still people have to be enrolled. There might have to be an initial physical exam that isn't going to work through telemedicine, but once they're enrolled, then you can use telemedicine-based approaches. You can ship devices to them that maybe they're wearing, or for collection of blood samples. And so one of the best examples we have from Merck for why at-home sampling we implemented for the first time, it was for a migraine study, right? And so you can imagine, if you're a migraine sufferer, do you really want to sit around a clinical site waiting to have a migraine so you can be dosed and we can take a blood sample from you? No, not at all, right? And so we developed at-home sampling for that trial. So we had people come in and train them on how to do a finger stick lancet and collect blood onto a paper card. And then we sent them home and we ran the study. So that's
awesome. That's awesome. And I agree with you. And I want to kind of tell my own story about what I totally agree with Kevin, what virtual means. And so, let's see. So I do a lot of my virtual visits to a doctor virtually. I do them via, and I know this sounds crazy to people, and some people say it's crazy and you can't trust it and you got to go in, and okay, that's okay. That's your opinion. Certainly if you're a healthy individual and you're not super, super sick after that first visit, almost everything can be done via telemedicine nowadays. And so I have a doctor that and I'm going to have one of them on my show here in a couple of weeks, but we do everything virtual. We text, there's an app, it's secure. He tells me how I am. He can even take some of my vitals from my smartphone, all those types of things. And then I also do at-home sampling. You can go to Walgreens and CVS and you can get your own lipid testing kit that will test your cholesterol, your HDL, your LDL, your triglycerides, all of that. So I don't visit a doctor to do that. I send it in and I get that information myself. I can read it just as easily as an MD can. And so I think the future really is virtual, and it empowers the patient. It really does empower the patient to actually take care of themselves, do their own testing, understand their own testing as much as they can. It doesn't mean that you're doing this cowboy and willy-nilly. You're still going to be directed by an MD. At the same time, it's like it's empowering the patient. And so that's what's, I think, is the future. I mean, do you think more and more virtual and telemedicine, that is the future, Kevin?
Yeah, absolutely. I think the interpretation is going to take a bit longer. You don't want to, and I think that's where some companies that have tried to promote testing if you're trying to make a diagnostic test, you have to be really sure, one, of the data quality, and then two, what the interpretation of that data means, right? And so, and is that an anomalous test? And do you have a follow-up test, right? Because you would hate for someone to say, oh, I did my blood sample and I see this and now I've changed something and it was not for a good reason. I think that'll come. I think we're seeing just even how medical interpretation diagnosis gets done, as all the things like electronic medical records get digitized, and the collective knowledge of human health becomes something that is, available, anonymized, personal identifiers taken away. But you can imagine your doctor hasn't seen every possible disease, in the world, and so his knowledge is based on his experience. Yeah. But if he could enter those symptoms or tests into an environment that would have a full, fully shared set of records, it's like, oh yeah, we've seen a few other cases of that have similar. Here's the possibilities, right? And so, yeah.
We actually have some programs like that in Idaho. It's called the ECHO program, and it's just kind of how the communities get together in this very large state, and MDs and healthcare professionals come together and talk about some of these things on a routine basis. It's been very, very helpful during the pandemic about, what kind of sick people are they seeing, what precautions should they take, and all those kind of things. And so that is only the future. I mean, we have the technology to share all this information. Obviously, patient confidentiality has to be the most important thing. I get that. And that can be easy to do. That can be something that's possible. So we have to think about the possibilities and not necessarily hold us back with all the risks. And so I love where this is going. I love that you've been a part of the virtual trials and I think that is the future, Kevin. Thanks for all the science that you've done, and how you've driven the communities, whether it be with microsampling, virtual trials, at-home sampling, time of flights, all these types of things, mass spectrometry, and so many things about science. And kudos to you. As we wrap this up, I've loved your story. That's the most important part of this. I just loved your story, and I can already see what some of these answers may be, but I always like to end with, everybody in life has something that they think they failed at, or somebody would maybe externally looking up on their life say, oh, wow, they failed at that. I wonder how that made them feel. Now, most of the people that look at people like that and think about failures are actually the people that aren't really the ones that are moving forward. They're the ones that like to, I don't mean to diss on anybody, but they're the ones that probably are paralyzed and not moving forward at all because they're afraid of risk. And so they've had no failures because they've really not accomplished anything. And to accomplish things, you have to go through ups and downs. And so, Kevin, tell us about one apparent failure that maybe you think's a failure, or maybe somebody else thinks it's a failure, but it really caused your life to be on a different trajectory for success. What would that one failure be?
I got a list.
We all do. That's what I'm loving. You can choose as many or as few as you want.
Well, I'll give you two. I'll give a quick one. Going back to university days when I said I wasn't really focused, and because I think people get hung up on books and book smarts, and so yeah, I had a bad semester, right? I had I'm admitting this in public. I had 4 Fs and a D.
Wow. Kevin, I want to tell you, we're in the same boat, but mine weren't that bad. Well, I'm actually surprised to hear that from you.
I ended up in the hospital. I had acute mono. I was anemic. I was messed up, right? And so, yeah. And anyways, but that was before that I moved to England. I came back and I became an A student, right? So, yeah, it was a lack of focus in life and overcoming that. And then the other one that's had a big impact, and I taught this migraine trial where we trained patients to go home and collect samples. We got that data, and it was horrible, complete mess.
Really?
Not because the analytical quality of the data was wrong, but we didn't understand a couple things. We didn't know when patients were taking their drug or if they're taking the drug, and we didn't know when they were collecting the sample because they were just recording it in a paper diary. And so there's, okay, how do we improve the quality of the sample and make it easy for the patient to collect, all the information associated with that? Because the more steps you ask a patient to do. So that's really driven, the focus of a lot of what I've been doing over the last 5 years is drive to make patient-centric sampling a reality. And I think we're there. But that failure is like,
awesome.
It's like, oh my God, that's horrible. I didn't realize all these things that I was totally unaware of. And so it just made me work even harder to solve those problems.
That is so awesome to hear. I think we found our 1 minute clip. I usually have a 1 minute clip for all of my podcasts to just kind of advertise. And that was a really good one. And what great stories. Those are both things that somebody could have just said, oh, well, I give up.
Yeah.
I give up. I'm going to stay in England. I'm not going to finish my degree. Or, wow, this data is just junk. I don't know what to do with it. And they get paralyzed by that. And there's obviously, I can only imagine both those scenarios, whether it be having to deal with your parents and what they're thinking of you when you're in England and dropping out of university, or having to deal with Merck and all your colleagues that the data is not what you thought it was going to be. And now you have to tell them, yes, but we can do it better. And you double down on your theory. That is not easy.
No.
That takes a lot of risk. And you know what it takes that a lot of people don't have, Kevin, is it takes courage. So congratulations. You've taken those risks. You have that courage. You have a positive mindset. You love what you do. Keep inspiring, my friend. It's just great to know you. So thank you so much. We're wrapping this up. Remember to see us on YouTube, on Spotify, on iCloud, on iTunes, and on SoundCloud. This will be recorded. This is all recorded, and it'll be on there in a week or so. Find my website, drsneedham.com. You can follow me on LinkedIn, on Instagram, and also on Facebook. And just thank you so much, Kevin. I hope I was on an airplane a couple weeks ago. I'm going to be on one soon again. One of these days we'll see each other in one of the conferences when we can all get on airplanes again. And so, Kevin, thank you so much. It's such an honor. It's an honor to know you. Great podcast. I'm seeing my phone's lighting up, seeing great comments already. So thank you very much. And you have a wonderful day, and we will be in touch very, very soon.
Great, Shane. It's been fun. It went by way faster than I thought.
Hey, you know what? We could probably do this for 4 or 5 hours, and maybe we'll do it again.
All right.
Hey, take care, Kevin. Have a good day.
Okay. Bye.
Bye.